Welcome to ePathshala. The topic that we are going to discuss today is materials and methods for encapsulation. Encapsulation is a process to entrap active agents within a carrier material, and it is a useful tool to improve delivery of bioactive molecules and living cells into foods. Materials used for design of protective shell of encapsulates must be food grade, biodegradable and able to form a barrier between the internal phase and its surroundings. If you look at the success of the encapsulation process, it is often linked to know-how of the formulation or of the chemistry to achieve stabilization. You see, this is especially in the case of food industry, where the number of acceptable materials is very limited. Encapsulation may therefore be defined as a process to entrap one substance, which is called as the active agent, within another substance wall material. We should understand that the encapsulated substance except the active agent can be called the core, fill, active, internal or payload phase, the coating, membrane, shell, capsule, carrier material, external phase or matrix. Now, in the food industry, encapsulation process can be applied for a variety of reasons. Encapsulation is a useful tool to improve the delivery of bioactive molecules. Example, antioxidants, minerals, vitamins, phytosterols, lutein, fatty acids and living cells like probiotics. You incorporate this into foods. In most cases, encapsulation refers to a technology in which the bioactive components are completely enveloped, covered and protected by a physical barrier without any protrusion of bioactive components. Also, encapsulation has been defined as a technology of packaging solids, liquids or gaseous materials in small capsules that release their contents at controlled rates over prolonged periods and under specific conditions. Produced particles usually have diameters of few nanometers to few millimeters. Now, let us look at the applications for modification of physical characteristics. Here mainly it is done in order to 1. Allow easier handling, 2. To help separate the components of the mixture that would otherwise react with one another, c. To provide an adequate concentration and uniform dispersion of the active reagent. Now is the time to look at the materials used for encapsulation. A lot of substances may be used to coat or encapsulate solids, liquids or gases of different types and different properties. However, regulations for food additives are more rigid than, for example, pharmaceuticals. Different compounds widely accepted for drug encapsulation have not been approved for use in the food industry because many of these substances have not been certified for food applications as GRAS materials. Actually, the whole food process should be designed in order to meet the safety requirements of governmental agencies. Let us look at the important criteria for selection of encapsulation material. Usually, these depend on the functionality that encapsulate should provide to the final product. Encapsulation, potential restrictions for the coating material, concentration of encapsulates, type of release, stability requirements and cost constraints. Materials used for design of protective shell of encapsulates must be food grade, biodegradable and able to form a barrier between the internal phase and its surroundings. The majority of materials therefore used for encapsulation in the food sector are biomolecules. Except to be natural, materials have to be providing maximal protection of the active material against environmental conditions to hold actives within capsule structure during processing or storage under various conditions. They should not react with the encapsulated material so that the good rheological characteristics at high concentrations are maintained and to have easy workability during the encapsulation. Among all materials, the most widely used for encapsulation in food applications are polysaccharides. They are usually the starch and metabolites like amylose, amylopectin, dextrins, maltodextrins, polydextrose, syrups and cellulose, which are commonly used. Now these come out from plant exudates and extracts like gum arabic, gum tragacanth, gum carria, mesquite gum, galactomannans, pectins and soluble soybean polysaccharides. Recently, marine extracts such as carrageenans and alginates are also present in foods. We should also understand that microbial and animal polysaccharides like dextran, chitosan, xanthan and gellan are now exploited. Apart from natural and modified polysaccharides, proteins and lipids are also appropriate for encapsulation. Examples of the most common milk and whey proteins are caseins, gelatin and gluten. Among the lipid materials suitable for food applications are fatty acids and fatty alcohols, waxes such as beeswax, carnauba wax, candelia wax, glycerides and phospholipids. Let's first look at what carbohydrate polymers can help in encapsulation. Carbohydrates comprise more than 90% of the dry mass of all biomass and more than 90% thereof of the carbohydrate polymers are polysaccharides. These natural homo and copolymers are composed of sugar residues and or their derivatives. Many native polysaccharides contain a small percentage of peptide residues remaining from their biosynthesis. Native polysaccharides are of enormous varieties. Moreover, they also form valuable sources for chemically defined materials, thus expanding their applicability and usefulness. On a total, the polysaccharides are of tremendous economical importance. Now let's look at the applications and role of starch and starch derivatives. Starch, as we all know, is normally a white powder. Depending on its source, it may be odorless and tasteless. It is insoluble in cold water, ethanol and most common solvents. Starch consists of two constitutionally identical but different polysaccharide molecules. These are essentially the linear amylose and the highly branched amylopectin. Both amylose and amylopectin are complex macromolecules with the capability of interacting intra and intermolecularly to form ordered structures. In starch, the amylose molecules are embedded in a physical network of amylopectin molecules. This diagram shows the commercial sources of starch, which is divided into cereal grains, roots and tubers, stems and pith. Now looking at the starch derivatives, the aim of starch modification is to alter the structure and which affects the hydrogen bonding in a controllable manner in order to enhance and extend the industrial applicability. Many functional derivatives of starch are marketed, including cross-linked, oxidized, acetylated, hydroxyl propylated and partially hydrolyzed molecules. This means that the cross-linking replaces a limited number of hydrogen bonds between starch chains by permanent covalent bonds. The next issue is with respect to maltodextrins. These are creamy white hygroscopic polysaccharide powders which are almost tasteless or only moderately sweet and easily digestible. Maltodextrin is an acid or enzyme catalyzed starch hydrolase. The most common dextrins refers to any product obtained by any method like heat, acid and enzyme for degrading the starch. Dextrin is also referred to as starch gum. As an example, as we all know, yellow corn dextrin is used to encapsulate water insoluble flavors and oils by spray drying. The more recent group of compound is the cyclodextrin, which are a family of cyclic oligosaccharides. Degradation of amylose, amylopectin or glycogen by diluted acids or by enzymes of example Bacillus messenceris yields these cyclic dextrins. Typical cyclodextrins contain 6 to 8 glucose monomer units. Next is the most common again, polydextrose, which is also designated as poly-D-glucose. It is a synthetic, highly branched polymer with many types of glycosidic linkages created by heating dextrose with an acid catalyst. Polydextrose is preferably used as a bulking agent because it is tasteless and it is similar to fiber in terms of resistance to digestion. Modified starch and maltodextrin have therefore been used for coating and encapsulation by several technologies, including spray drying, fluidized bed spray drying, fluidized bed granulation, compacting, and most importantly, extrusion. Molecular inclusion is possible with cyclodextrins. Maltodextrins are cheaper than gum arabic and are therefore in some cases recommended as a partial substitute in encapsulation. Cellulose and cellulose derivatives are the next group of compounds that needs attention. Cellulose is a major structural material of plants. The primary cell wall of green plants is made up of cellulose. Wood is largely cellulose and cotton is almost pure cellulose. Other important natural sources are flax, hemp, jute and straw. Biochemically, cellulose is a polymer of beta-D-glucose. Due to the absence of side chains, cellulose molecules can arrange close together and form rigid structures. In contrast to starch, cellulose is much more crystalline. Whereas starch undergoes a crystalline to amorphous transition at 60 to 70 degrees in water, cellulose requires 320 degrees centigrade, which is high, to be amorphous in water. Looking at the derivatives of cellulose, the hydroxyl groups of cellulose can partially or completely react with various reagents to produce derivatives with properties desired and required for food applications. Methyl cellulose is a hydrophilic white powder which dissolves in cold water, forming a clear viscous solution. Abbreviated as MC, this has a good film forming property and to a certain extent surface activity. However, it is not digestible. Plant exudates and extracts comprise polysaccharide plant exudates and extracts, which are complex macromolecular substances. Some of them are mixtures consisting of oligomers and polymers of different chemical structure and chain architecture. Several of the materials used for food encapsulation are products of guminosis and also therefore named plant gums in practice. Few plant species are cultivated at present to provide gums for the food industry. Most of them belong to the leguminoso family, which are the pulses. Some examples of plants with appropriate gum harvested thereof are listed below. Acacia senegal, which is gum arabic, astragalus species, which is gum tragacanth. We have another Stenoglobose, which is a gar gum, and a Ceratoni silica, which is a locust bean gum. Finally, we have marine extracts. Seaweed can be regarded as another almost unlimited source of different types of polysaccharides for many industrial applications. Some of them have been found to be useful for encapsulation in the food sector. Carrageenans are prepared from red seaweed, and seaweed selection, processing and blending of different extracts can control the variations of the carrageenan types. Alginate are generally produced from marine brown algae, although they may be synthesized as an exocellular material by a few bacteria. Microbial and animal polysaccharides. Polysaccharides produced biotechnology by bacteria are biopolymers with novel and partially unique functional properties. Examples which are interesting for food applications are xanthan, gellan and curdlan, though the WHO has at the present time certified the latter only for a few countries. Proteins are natural macromolecules composed of linear chains of amino acids. Of this, gluten proteins are strongly associated with baked products. However, the unique properties of wheat proteins have stimulated an interest in their use for other applications than traditional ones. It is likely that the range of proteins recovered from milk will extend in the future. Tailoring of milk protein products to meet specific functional requirements for individual applications will become increasingly important. Hence, the use of milk protein products in emerging technologies such as the manufacture of edible films and the micro-encapsulation of ingredients will benefit from further development. Gelatin. This is a heterogeneous mixture of single or multi-stranded polypeptides. Gelatins in their pure form are translucent, brittle solid substances which are colorless or slightly yellow, almost tasteless and odorless. Gelatins melt when heated and solidify when cooled again. On warming to 35 to 40 degrees centigrade, the gels will once again dissolve. Whereas from dilute solution, gels with a melted mouth texture are obtained, the gels from higher concentrated solutions exhibit elastic gum-like textures. Gelatins do not occur naturally, but are manufactured from collagens by processes that destroy the secondary and higher structures of collagen. The latter is the major constituent of all white fibrous connective tissue occurring in animals. The next macromolecule is lipid, which is characteristic of their insolubility in water. They are hydrophobic. Lipids involve molecules and substances of large diversity and structural variety such as oils, fats, waxes and phospholipids. Triglyceride is the main constituent in animal fats and plant and vegetable oils. Most natural fats contain a mixture of different triglycerides. The commercial source of mono and diglyceride is the same as for triglycerides. After the macromolecules, the other components include polyvinyl pyrrolidine (PVP), which is a synthetic neutral polymer soluble in water and organic solvents. The good film forming ability makes it an excellent polymer for coatings, which requires good temperature stability. Paraffins is a white, odorless, tasteless, waxy material which is also available at food grade quality. Paraffin is edible but not digestible. Looking at inorganic materials, there are several food grade inorganic materials that have been described as being useful for coating or micro encapsulation in food applications. They can be utilized alone or in combination with other materials. They include tripolyphosphate, silicon oxides or aluminum oxides. Now, looking at the methods of encapsulation, there are a number of techniques which are currently available for encapsulation of food components. Drying is commonly preferred technique as most of the components are prepared in the liquid form. Techniques like spray drying, spray bed drying, fluid bed coating, spray chilling, spray cooling or melt injection are used to encapsulate active agents. Many encapsulation processes are based on making first droplets of the active, and these droplets are in gas or liquid phase via different physiochemical processes. There exist many classifications for encapsulation methods. The easiest classification may be based on the equipment needed to perform the encapsulation, which is shown in the following figure. The first one is spray drying, which is one of the oldest approaches for producing capsules. The small droplets are either cooled down or dried. This use of spray drying is largely developed in the food industry, as the process is very similar to the one employed to produce classical food powder, more commonly in milk powder. The technology allows large production. Spray drying of active agent is commonly achieved by dissolving, emulsifying or dispersing the active in aqueous solution of carrier material, followed by atomization and spraying of the mixture into a hot chamber. The picture of the spray drying process is seen in this image. During this process, a film is formed at the droplet surface, thereby retarding the larger active molecules while the smaller molecules are evaporated. Spray dryers in food industry are usually atomizing the in-feed with a high pressure nozzle or centrifugal wheel and operates with a co-current flow of air and particles to give a minimal overheating of the particle. The size of the atomizing droplets depends upon the pressure drop across the nozzle and the velocity of the spray. The size of the atomizing droplets also determines the drying time and the particle size. The temperature of the droplet surface corresponds at any point in the dryer to the wet bulb temperature of the gas phase surrounding the droplet as long as the particle surface is wet. By the time the droplet is controlling the air inlet temperature, typically 150 to 220 degrees centigrade, the flow rate, the feed temperature and evaporating cooling, it should be ensured that the droplet temperature never exceeds 100 degrees centigrade. This is very important because this temperature might be indicated by the air outlet temperature, which is typically 50 to 80 degrees centigrade. The larger the spray dryer, the longer the residence time of the particle in the dryer and hence the larger the maximum size of the droplets that can be dried. Atomizing nozzles are usually mounted to spray downward, but it is also possible to spray upward like a fountain, which permits somewhat larger droplets to be dried because of the larger residence time of the droplet. During the drying process, a film is formed at the droplet surface and the concentration of ingredients in the drying droplet increases. Finally, a porous, dry particle is formed. The carrier material used should meet many criteria such as protection of active material, high solubility in water, molecular weight, glass transition, crystallinity, diffusibility, good film forming properties, good emulsifying properties and low cost. Examples include natural gums, proteins, carbohydrates and lipids. Conventional spray dried encapsulates release their active agent immediately upon addition to water. The second process is dripping. This consists of extruding droplets from nozzle in gentle conditions. Droplets may be solidified by cooling or by jellification. Productivity may be increased by forming a liquid jet and breaking it into small droplets. They may multiply in the nozzle or they start working with spinning devices. The main advantage of this technology is the low sized dispersions of the microcapsules. The main drawback, however, is the productivity. The third process is that of emulsions, which is a thermodynamically stable two-phase system, and both the oil and water phase will separate. Emulsions can be used as delivery vehicle for either water soluble and lipophilic vehicle for agent as an active agent in food products. There are two considerations that must be taken into account when formulating an emulsion for controlled delivery. First, the emulsion system must be stable right up to the point of application. Second, on application, the emulsion should behave in a manner consistent for achieving the desired delivery. The next one is spray coating. This is not to be confused with spray drying because this process consists of fluidizing a powder in a fluid bed or a pan and spraying a coating solution on the fluidized particles. The coating is solidified by drying or cooling. This is the most promising method in terms of performance and flexibility, but costs have to be significantly reduced. Particles may also be suspended in a solution. Interaction between particles and the solution result in the formation of a coating layer around the particles. Next, dispersed emulsion droplets may be turned into microcapsules by different processes. In the food industry, the main method used is called co-azeravation, which consists first in dispersing an oil phase in a polymer solution and then inducing the precipitation of the polymer at the interface of the droplets. If the productivity is very good, this method has an important drawback, which is cross-linking with glutaraldehyde, a non-food grade molecule which is necessary most of the time to obtain stable microcapsules. Fluid bed coating is another encapsulation technique where a coating is applied onto powder particles in a batch processor or a continuous setup. The powder particles are suspended by an air stream at a specific temperature and sprayed with an atomized coating material. The coating material might be an aqueous solution of cellulose or starch derivatives, proteins and gums. Extrusion methods consist of dropping droplets of an aqueous solution of polymer and active into a gelling bath, which is some kind of a calcium chloride solution. The dripping tool can be simply a pipette, a syringe, a vibrating nozzle, a spraying nozzle, jet cutter or atomizing disc. Several constraints make the development of encapsulation process difficult. First, encapsulation is an extra cost which has to be minimized to be economically acceptable. This applies to the materials used to build the microcapsules, but also to the equipment or processing conditions. The number of food grade materials suitable to form microencapsulation is also very limited. Consequently, the food engineer has to play finely with the coating or membrane formulation to achieve appropriate and specific properties. Developing an encapsulated product remains a challenge requiring a multidisciplinary and integrated approach. Many encapsulation technologies currently exist, but many of them are still at the development stage. Once fully tested and validated, these new methods will undoubtedly broaden the spectrum of possible applications for this versatile technology.